Okay. Next week, where are you going to be?
At home.
At home, wherever suits you. Totally up to you. Sorry? Cocktails, right. Right, let's go to cocktails. And which are you going to log into? Which web server, web page, browser? Doesn't matter, suits you. You're going to go into views, aren't you? Yep. Views, what thing are you going to go into in views? Q&A tutorials, right? Be wherever you like, be on your phone, totally up to you. Q&A tutorial. You'll see a shared screen from me. You'll be able to type in questions. I'll be recording that Q&A tutorial. When is your report part A due? Where's my colleagues? Oh yes, what date was that again? 13th, what time? 9 a.m. 8 a.m? 9 a.m?
No, it says on here 8 a.m.
8 a.m. indeed. Why 8 a.m? Because I get to my office about 7:30, 8 o'clock. I'll download everything, give it to the external examiners, and bam, off we go. After that time, there'll be late submission penalties, won't there, unless you have a special consideration in place. That report, part A, how many words? 1,000. Now, we don't ping you for being too much over, a couple of hundred, no big deal. A couple of hundred, well, did you cover the topic area? What headings are you going to use? Research, diagnosis, treatment and policy. Now in the Q&A I'll go through some of that stuff as well. You must have those headings, is that clear? They must be clear headings. You can have subheadings, it's up to you. I'll discuss that again next week. People are asking about references. Yes, you must be looking at current references as much as possible, but some go back to the 50s, some beyond, depending on the disease state. But if they're all from the 50s and 60s, that's not going to cut much mustard, is it? Particularly because a lot of these disorders can be diagnosed with things like PCR. Was that developed in the 50s and 60s? The guy who invented it was invented in the 50s and 60s, but not the actual process. Indeed, I got to meet him. He was in San Diego. He was a gene jockey trying to work out how to melt DNA, found a beaker, put the tube in, melt the DNA, put an enzyme in, and he just kept doing that by hand, and he had PCR going. Then he went down to the beach in his convertible big car with a surfboard and went surfing. That's what you do in San Diego, except the water's a bit cold. So you're going to have 1,000 words. What kind of referencing format are you going to use? Vancouver. You do Harvard and you'll lose some marks. You do Vancouver. Vancouver is what kind of numbering system? Where does the number go? The superscript, thank you for correcting that. Superscript. Where does it go? It goes right behind you where you make a statement that you've got from somebody else. That doesn't mean a quote. I don't want to see reports where all I see is quote after quote after quote, because it's not your report then, is it? You have to interpret what someone's saying, put it in your own words, and then you put the reference. Again, I'll cover that in the Q&A. Told your colleagues at Parramatta and Hawkesbury last week to remind them that Q&A is next Tuesday evening, this time frame, at home with cocktails, feet up, on the phone, listening to what's going on, writing your report. Is that a good time? That gives you almost a week and more, doesn't it? Or roughly a week-ish. If I went into Wednesday afternoon, it's less time, isn't it? Thursday morning, the Hawkesbury time. Yes, you can do that. You can do all three. It's up to you. It's recorded. And after that particular date, before the submission, you'll get a response from me. I need clarification. The response will be watch the recording. Is everyone clear on that? Because right now I'm doing lots of COCs and lots of other postgrad stuff, and we're really busy for the next couple of weeks. I mean, really busy. We have papers to get out. The other thing you need to worry about is you should have been doing some research already, shouldn't you? That would be wise, because when you go to the Q&A, that's where you seek clarification, not how do I start? What referencing system do I use? What headings do I use? Look at all the documentation. You're wasting time, okay? I booked 2.5 hours for each of these Q&A tutorials. You'll click on it, big purple circle will go by, eventually it'll come up. If it doesn't work first time, change your server or provider, whatever system you're using. I'll start about 5-10 past the hour, start recording. Is there a question over there? That's all cool? Okay. So make sure when you come to it, you've got some real question I can deal with, okay? Yes, ma'am. Is there a quiz next week? What do you think? Yes. Wasn't there a couple of catch-up quizzes? Go yes. There's a total of 12. The first one in week 1 was a practice. There was no marks to it. So next week, yes, there will be a quiz. Do you need to be at university to do it? I could be mean, but I took a happy pill the other day, so no, you don't need to be at university. It won't have any IP restrictions. You'll do it wherever it suits you. The same deal, you'll get a quiz. You'll get a password to log in. I'll tell you that at the end of the session next week. And it doesn't matter what session you're in. So if you're a Parramatta, Hawkesbury, Campbelltown student, you can go to any session, multiple sessions, doesn't matter. But there is a quiz next week, Thursday at 1 o'clock. You'll get access to the will assay ready for your mid-semester exam. When does that start? Next Tuesday. When does it finish? Friday, 9 till 5. You could do it at midnight, it's up to you. Now, you can almost guarantee IT are going to do an update of views during that process. Be aware of that. So if you do it during the night time from about 11 o'clock till 4 a.m, I won't be able to help you. And even between 5 and 6, I'm usually running. So you'll have to wait till I get to my office about 7:30, 8 o'clock. That'll be up on views, notices to remind you of all of those things. Quiz, report, Q&A, mid-semester exam, and the will assay for the mid-semester exam. So I'll put that up on Thursday. You need to find it. You'll need to do the calculations.
The mid-semester exam, where will it come under that assessment?
On the left-hand panel in views, the black thing that slides out with Q&A tutorial, you'll see it right there. You click on it, it'll come straight up for you, okay? Yes, mate. And the more you fiddle with it, the more you won't be able to stop it. You know that, don't you? Don't break a computer, it's not worth it.
Louder, please.
Don't break a computer, it's not worth it. Okay? Done now? Cool. It's not embarrassing at all. Okay? Question was, the will assay, how's it going to work? What you will see, and I'll put a notice on views in my grades, your my grades, on views it'll have a patient number. You've had patient numbers all semester, you'll get a unique patient number, up to nearly 400. So you might have any number, 1 to 400. What you will then do is log on to the data. One of the questions will come up randomly. There it is, the will assay. You've already got the kit, the protocol, the bulletin, yes? You've read it, so you know what's going on, because it's not quite the same as the others, which means you've got time to look at it from Thursday, 1 o'clock. During that particular mid-semester exam, you'll get data like we're giving you, not just one strip with 3 data sets on it. You'll actually get a whole suite of data sets with your patient number on. You find your patient number, you'll have the standards, like we always do to you, and the unknowns. You use those to calculate a standard curve, and you work out the unknowns for your patient. On views, it'll give you, much like I've asked my supervisors, what's the range for your patient, A through F, G, H, whatever it's going to be. Is that kind of clear? The units will be there as well, so you'll select that. Just follow the instructions. Kind of clear on that? I'll put a notice on view to remind you Thursday. Yes, mate.
Can I log more than one time to quiz once?
Sorry?
Can I log in more than one time to the quiz?
You can try, but you only get one bite of the cherry. And like all of my questions, seeing how I took that happy pill after I made up the mid-semester, you get one bite at each question and no going back, must go to the next one. You've got 2 hours. 2 hours, how many questions? Not too much cocktails. Look at what I put, I'll discuss in the Q&A next week. That is double the time you need to do it, double the time. Everybody should be doing it in an hour-ish. And I don't mean me, I mean you guys should be able to do it in an hour, if you're organised. Now, it's an open book. It's called a formative assessment, not a summative. Summative, invigilators, don't copy, don't cheat, don't do anything. Formative means, like your colleagues have done in the past, get together, discuss, do things, whatever, use Zoom, help yourself because it's going to challenge you, I'll guarantee it. If you know the language of biochemistry, you better do this really well. If you don't know the languages we've been talking about, it's going to remind you, you want to get on top of it pretty quickly because the final exam's coming at you, isn't it? Very fast now, isn't it? We're almost over the hump, mid-semester. Yes, mate?
Are all the questions in the mid-semester just based on the will assay, or is there other general questions about what we've learned thus far?
I'm glad you asked that question. I'll remind you again in the Q&A. The mid-semester exam is based on everything you've done, including all the will assays, all the lab activities. There's one calculation, a will calculation for the new assay I'll give you on Thursday at 1 o'clock. So everything you've done is up for grabs. Is that kind of clear? All the stuff in the lab, the things we've been discussing in the workshops, those are up for grabs. It's all totally random. 600 plus questions, they're going to come at you, and a whole suite of them. And everybody's randomly different, which is a good thing, isn't it? Yes. Any other questions before I get to the serious stuff? All right, let's get serious. Last week we talked about glycolysis, which is the left-hand panel. Look at the left-hand panel. What do you see? Glucose, hexokinase, phosphofructokinase. Which acronym are you going to put next to that? PFK1. I run down, you see all those double-ended arrows. I get to the bottom, what do I see? Pyruvate kinase. If your liver, doesn't the liver do gluconeogenesis when it needs to? On the right-hand side, that's the comparison. You notice the double-ended arrows are all the same. We can go backwards and reverse those reactions, no big deal. But any single-way arrows, we've got to have some bypass reaction. Let's start at the top. Hexokinase does what? Traps glucose. Traps it, activates it to do something with it. If I'm going to go from glucose 6-phosphate back to glucose, I'm going to show you today, a bit complicated but expensive energy-wise, I'm going to have to use this thing, glucose 6-phosphatase. There's a surprise, take the phosphate off. Got to do that in the ER. That's to get back to glucose. I run down here, what have I got here? PFK1, are there a number of steps in between? Yes, there are. PFK1, if I want to bypass that because that can't go in reverse on this one, fructose 1,6-bisphosphatase. Makes sense? Kinase phosphorylates, phosphatase takes it off. You kept telling me that. I run down the bottom of glycolysis. I've done all the split. I can put it back together. I get down to the bottom. They're all two-way arrows. What have I got here? PEP. Isn't that the will assay you have to do? Then, to bypass pyruvate kinase, I need two steps. What's this guy? Oxaloacetate. What's its acronym? Write it down. Remember, that's part of the citric acid cycle. I need a couple of steps. This pyruvate carboxylase using ATP to make OAA, and this low-energy carrier, GTP, a couple of those, with carboxykinase, PEB kinase. What am I doing? Putting phosphates back on. Where are we doing this? Which cell type? Not quite, not in the mitochondria. Which cell type? Not organelle. Hepatocytes. Please write it down. It's in the hepatocytes, in the cytoplasm of the hepatocyte. We haven't entered the mitochondria. That's what we're going to do later on today. Please note there are two ATPs going in, two GTPs going in. Why is that? Exactly. You just do the opposite. Then you need two pyruvates to go back up to make a glucose, don't you? Think of one glucose molecule, two pyruvates, one glucose molecule, I need two pyruvates to make it. So it's very energy expensive. This process, and I want you to underline this, is reciprocal regulation. In your liver, and this is what we've got to think about today, how do you regulate this and why? And again, think of the final exam. It won't happen in the mid-semester where you have to write small essay responses. It's about the fed, fasted and stressed state. In the fed state, you're coming down, aren't you? The fed state, you're making fat, you're doing various things. In the fasted state, you're more than likely going back up, aren't you? What are you doing? Making glucose. Who controls that? Glucagon. Going back up, is that also the stress state? Yes. So that short answer question, you know there's 4 of them, you know one of them's going to be that one. What's happening in the stress state? We're going back up gluconeogenesis to produce as much glucose as possible. Now today we're going to talk about some of these enzymes, and the process we need to keep in mind is how does the liver regulate this, and how is it different to the liver and muscle cell activity? Because what does a muscle cell do? It's got to use energy. Energy for work, to run away or fight, whatever the case is. In this case, the liver's going to be under the control of what? Glucagon, adrenaline, for example. So we have to run back up there, generate glucose, send it out really quickly. So please underline this reciprocal regulation. How do I do it and why do I do it? Well, today we're going to see you need to regulate some of these enzymes, like these characters down here, from a gene level. Think about if I'm constantly fasting, I'm not eating a lot. What am I telling my liver to do all the time? If my glucagon's up all the time, what's my liver being told to do? Glycolysis or more gluconeogenesis? Which one would predominate? And the more you fast, the more you drive the genes to make the enzymes, like these characters, to make glucose. The same the other way. If I'm constantly eating, like, all the time, what am I doing? Constantly sending this system down. What are you doing? Making fat mostly. We're comfortable with this concept. Okay. Keep in mind, 3-carbon pyruvate. We're going to run down here from PEP, pyruvate. We're still not in the mitochondria. We have to do that later on today and after the semester break. And incidentally, the mid-semester exam only goes up to today, and the will assays, all that, nothing after that. Let's move to the next one, shall we? This pyruvate kinase. How is the liver going to regulate that? It's a little bit complicated. This thing is rather big. What's a tetramer? Not a tetra pack, a tetramer. 4 subunits. So it's more than likely an allosteric enzyme, isn't it, with 4 active sites at least. Pyruvate kinase is a tetramer, 57 kilodaltons, so that's fairly large. It's found in liver and muscle, but it has isoforms. What does isoform mean? They covered that in FP&G, didn't they? Yeah, it is essentially a variant in the enzyme. They essentially do the same thing, but each tissue has a slightly different variant to do a slightly different job. Not the actual enzyme activity, but how you regulate it. F16BP, who was that? Let's go back a slide. Where's F16BP? F16BP, write it down now so you don't forget it. F16BP activates both isoforms, that is the muscle and the liver isoform. That's a question for you. Why? Why would that happen? Let's go back. Look at it. F16BP builds up in the cell. Why would you activate this kinase? What's happening to the cell at that point in time? What's it experiencing? Sorry? Absolutely. But also, what's happening to your liver? It's making fat, isn't it? It's burning through the process of glycolysis. What's your muscle cell doing? It's also storing glycogen. It may be working as well. The next bit, ATP and alanine allosterically inhibit both forms. What does that mean? Let's go back a slide. ATP, put whether you see ATP, square bracket, arrow up, that's the cars building up in the yard again. What's that telling the cell? Do you need the currency of energy as ATP? No, it slows it down, doesn't it? So what about alanine? What does that tell us? We've covered alanine right in the first lecture, the overview. I've hinted at it a few times. It's a small 3-carbon molecule. If that builds up, what does that tell in the cell? My wife's back from the US, so I'm going to have to leave on time today. So you need to hurry up. Otherwise, I get in trouble. Alanine builds up in the cell. What does it tell the cell? What's the cell experiencing? Remember, it's a simple amino acid. And if a simple amino acid and a common amino acid builds up in the cell, what's the cell not doing? Depends on the cell. What's alanine involved in, apart from the alanine cycle? Yeah, used to create what? Starts with a P. Proteins, thank you. If I'm not building proteins, what am I doing? I'm not busy. Why the hell are you making more energy to do work if you don't have any things to do? You've got too much alanine hanging around. You're not making things with it. Gives you an indication of what the cell's going through. What you're seeing here, alanine is the signalling for abundant building blocks. If it builds up, you've got lots of bits to make cars. But I'm not selling cars, so what do? Stop making cars. That's the issue for the cell. The real control comes down here. The hormonal control of the liver isoform stops liver from consuming glucose when it's needed by the brain, the muscle, and erythrocytes. Let's look at the diagram. The left-hand side, underline liver only. You see the dashed line through the middle there. So here on the left-hand side, glucagon, green triangle, tells what? Tells this enzyme to get busy, phosphorylating, pyruvate kinase, I've inactivated. What's the glucagon actually telling that enzyme to do? Let's go back a couple of slides. Pyruvate kinase is inactivated in a liver cell that's experiencing glucagon. When do you see glucagon go up in your tissue and body? Blood circulation. Fed, fasted, fasted. So what is that telling your liver to do? Make what? Glucose. So what should you not do? Keep making pyruvate, right? Okay. So now what I've done is I've inactivated. This thing, PP, what do you think that might stand for? We saw one last week, PP1. Phosphatase is right. What's the first P stand for? Yeah, protein, pyruvate, phosphatase. So what you're doing, taking the phosphate out, now you've got an active one. You see there, L and M, what do you think that stands for? L, sorry? No. Liver. What's the other tissue we're worrying about here? Muscle. Write it down so you don't forget. So now I've got pyruvate kinase in my liver and muscle busy doing what? Taking PP through the system to pyruvate and... Is that right? Right. So I'm making pyruvate. Does that enter now the mitochondria? It might. With an enzyme we're going to see PDH. But something like alanine, if it builds up, it has a negative impact, doesn't it? It starts to shut this process down. ATP, acetyl-CoA, long-chain fats also have a negative impact. What does that mean? They're building up, the cell's not sending them out. So you're shutting down the process. You're telling the cell, I'm too busy. I've got too many cars on the lot, stop sending me stuff. Not going to happen. F16BP says go forward. Let's go back a slide. F16BP, I've got lots of it, run through it. So I've got incoming glucose, do something with it. If I'm a muscle cell, store it as glycogen. If I'm a liver cell, store it as glycogen or turn it into fat, whichever one's required. We're kind of comfortable with what that pyruvate kinase does. Okay? Isoforms. Liver has one isoform, muscle has another. They do the same kind of job, but the liver is hormone-sensitive kinase. You need to write that down. The liver pyruvate kinase is hormone-sensitive. Why? Because when you're fasting, your liver has to make glucose. When you're fasting, does your muscle make glucose? No, write it down. So that needs to be why there's different isoforms, which we see lots of ways to regulate key regulators. We comfortable with that concept?
Can you explain the last concept one more time?
Sorry?
The last sentence that you just said.
Yeah. So that, for instance, when we're in the fed state, what's your liver supposed to do? Make fat, make glycogen. In the fasted state, what's your liver supposed to do? Make glucose. Who tells the liver to do that? Glucagon. To control blood sugar, we'll see after semester break. Does your muscle make glucose? No, write it down. Muscle does not make glucose. It stores a bit of glycogen, it burns fat, but it doesn't make glucose. But both of them have to use this pathway, both of them have to use this pathway because it's glycolysis. They're going to make pyruvate. But the liver is under hormonal control. As soon as glucagon's up, we turn off glycolysis and go back up gluconeogenesis. So when you discuss that fed, fasted and stress state, you need to keep that in mind. Okay, we done with this one now? Okay. Let's have a look at this pathway. There you see a lot of steps missing, but the key steps and key regulators. Glucose, hexokinase, we'll come back to that character shortly. F6 phosphate down to F16BP, just jot it down so you keep remembering that. Who stimulates that pathway? Remember F26 last week? Was that part of glycolysis? Who made that character? BFK2, good job. Who inhibits it? Too much ATP. Don't keep making energy. Too much citrate. You should be able to work this one out. Why too much citrate would slow this pathway down, do you think? What's coming after glycolysis? Citric acid cycle. Too much of that means we're not running around fast enough. So we're going to feed back. And that's what this is, feedback inhibition. What about too many protons? Things like NADH, NADH+, NADPH. Too many of those means I can make free radicals. We're going to see a bit today, a bit of an issue, particularly in tumour cells. There was our key regulator we saw last week. We get down to this one. F16BP, a lot more steps. We get down to this guy, PEP, write it down. Who stimulates that? F16. Who inhibits it? ATP alanine. We've just discussed that, haven't we? Simple way to keep it in mind. Okay, let's go back up gluconeogenesis. We got to pyruvate, made a decision. Here comes glucagon. Okay, make glucose. I need this enzyme, pyruvate carboxylase. Here it is here. It's taking bicarb. Look at the screen. Bicarb, there's our acid function. 1, 2, 3 carbons of pyruvate. What did it make? With pyruvate carboxylase, circle the ATP. How many of those do you need? 2, if you're going to make glucose. Biotin, seen biotin before. Remember that swing arm, takes a carbon, transfers it somewhere else. What have we done? Transferred it onto pyruvate to make OAA. That's pretty straightforward. That enzyme just works away. There's OAA. Then OAA's got to be converted back to PEP. Now this is where it gets a bit messy. Phosphoenol pyruvate carboxykinase. Ugh. I think I need a drink after that of water, which is in the car. This guy is different. This guy is promoter controlled. Why the hell is that? Let's think about being the stress state. If you're constantly stressed by me and metabolism and everything else, what are you doing? What are you experiencing? Heart rate. What hormone is stimulating mostly? Acutely rather than chronically. Adrenaline. You're going to see later on, adrenaline stimulates gene expression. If I'm constantly stressed, I'm going to activate all the enzymes to do gluconeogenesis. Why? Because you're constantly getting the signal in your liver to make glucose. And that happens a lot here, because here, this thing is promoter control. The more I do stress, the more I fast, the more enzyme I need, this thing, phosphoenol pyruvate carboxykinase, to make glucose, because everybody else is there, aren't they? But this guy, if I'm busy fasting or being stressed all the time, I make lots of glucose. So I need all the machinery. My factory gets bigger and bigger to make this process work. Does that kind of make sense? So fed, fasted, and now stress state has an impact of driving more gluconeogenesis. Which cells, which tissue? Muscle or liver? The hepatocytes, write it down. The liver's going to make more glucose, and you'll see that constant adrenaline stimulates that. That's why people who are constantly stressed acutely, often you see them a bit kind of jittery. Why? Because their blood glucose is always coming out. Who encourages that pathway? Acetyl-CoA. Why is that? Put a square brackets around it, arrow up. If it builds up, it's not turning into fat or ketone bodies. What are you doing? Speeding up the process of going back to form glucose. What slows it down? ADP. What does that mean? Well, if you're not having enough energy currency as ATP, it means your pockets are empty. You've got ADP or AMP. What are you going to do? It doesn't matter if I'm getting a signal from glucagon to make glucose, but if my energy currency is low, and this is an energy expensive activity, then you're not going to make glucose. You're going to go away and make some more currency. Does that make sense? Because if I don't have the energy to do the job, I've got to stop doing the job and get more energy. Make sense? Indeed, write it down. What about up here? The bisphosphatase. Who slows it down? F26BP. Remember we saw last week? What did that do? AMP really slows it down. What does that mean? If here you didn't get the signal good enough, by the time we get up there, it's like you're really broke. You know, stop whatever the hell you're doing, make ATP because you won't be able to do anything. You'll be a dead cell. Who stimulates it? Citrate. Lots of it building up in the cell. Again, square brackets. Think car yard, build up, slow down the system. Everyone comfortable with that concept? Good. You'll go to pass and get some help. Okay, going to move to the next one. Okay. This reciprocal regulation is critical for your liver all the time. During the night phase, when you eventually go to bed after playing on your phones and computers, whatever silly games you play, which might be 1, 2 in the morning, how many hours sleep should you normally get? 8 hours should be the norm. Some people need more, some less. I know I need a lot of beauty sleep. In this particular case, we have this regulation of this stuff through the glycolysis, which is predominating. If AMP is so low, I need to generate energy. Gluconeogenesis predominates if AMP is that low. Let's have a look at this pathway here. Gluconeogenesis. What's the red cross mean? Shutting down. Green triangle means get busy. PFK1, get busy. PFK1, citrate, shut down. And you'll notice over here, AMP shuts down the FBPAs. So just look at the screen for a moment. Over here, these characters stimulate the glycolytic enzyme. Those don't affect so much this character, which is the gluconeogenic, but AMP definitely does. So up here, FBPAs, these guys don't do anything to it, but AMP definitely does. It's like the last thing that you get the message text from your parents or the bank manager that says, no money, no buy goods, no buy chattels, no buy lollies, no buy chocolate, no petrol, no nothing. Go back and do some work. Does that make sense? That's kind of the last stage. You will stop at that point, and then you will drive glycolysis. Here it's like, well, I'm still thinking about it down here, but I've still got a bit of ADP. But when I get to AMP, no, nothing left. We're ready for the next one. Now this schematic is a little bit hard to kind of get your head around for a start, but it's actually pretty straightforward. You see up there it says tissue-specific cooperation between glycolysis and gluconeogenesis. I've already talked to you about the liver and the muscle. Here is, kind of an ugly brown cell. That ugly brown cell has some glycogen storage. Who's that? Muscle maybe liver. Glucose coming in, there's it trapped. What did you do? You made pyruvate via glycolysis. If pyruvate then is being used to make energy, put over here electron transport chain or ETC. I produce CO2. That's where you cut carbons. PDH, I want you to write that down there, capital PDH here is going to take that into the mitochondria. Pyruvate can produce lactate. We'll see that in a moment. When is that occurring? Which cells? Muscle when it's really working hard, and sometimes red blood cells when you're not taking in enough oxygen. Why would a red blood cell worry about that? Does it run with mitochondria? Not when they're mature. So it's relying totally on glycolysis to stay alive, to do all the pumping. What about down up here? Let's go back up here. There's glucose. That stuff there with the little line looks like a maze. ER stands for what? 1 second. We're going to see in a minute we have to take glucose into the ER, take the phosphate off it, and then try and ship it out of the liver cell. Note that's the liver. But also note the kidney's involved. And I want you to put a little footnote. The kidney only gets involved when things get desperate, really desperate. And we'll see that with diabetes later on. Down the bottom, and I'll come to your question in a minute, down the bottom you see lactate coming back into the liver. And again, the kidney. Put down kidney when you're really desperate. Lactate's coming back in with that lactate. We'll see what it looks like shortly. We can make amino acids or we can make pyruvate, depending on what the individual cells are going through or you're going through. Now, I've said many times at the beginning of the semester, you don't turn fat, and I want you to write down fat as free fatty acids, long-chain fats. You don't turn those into glucose. What do you turn into glucose? Think of triacylglycerols. That's not going to be a glucose. That's not going to be a glucose. That's not going to be a glucose. What is? The glycerol. 2 of them, you turn into glucose, don't you? Fats not turn into glucose, but glycerol does. Animals, are we animals? Definitely are. Especially when there's food on the table. Yes, mate, you had a question?
My question was, if you're looking at laser pointer, you're pointing at the group.
Which one, sorry?
Just explain again why, down the bottom left-hand corner.
Yep.
Can you just explain again what happens when the muscles aren't working hard?
Okay, let's get you to get up and run over to the parking lot, other side of building 21, and then run back again. Keep on running. What happens in your muscles if you're not very fit? Fatigue. And what do you feel? Lactic acid buildup. What happens? The muscle cramps, doesn't it? So that lactic acid buildup is because you're busy working the muscle, not enough oxygen coming in to run through the mitochondria, do everything else, we'll see in the next couple of weeks. So you build up lactate. I want you to write it down now, lactic acid. I'll show you that shortly. Lactic acid builds up. Now that that's built up and I have a cramp, you're stuck halfway, what are you going to do? That leg's just cramped up. Slow down. What often happens? They try and stretch the muscle, but your recovery, that is, move the lactic acid back to the liver. Here it is, back in the liver. What does it do? It says, okay, you've just spent all that energy. You can't get back to your car or back to the lecture theatre. Now you're going to convert it to, well, if you were resting, you might put it into amino acids. If you're not, and you need glucose, and you're stressed because I'm saying get back here, you're going to turn it into glucose, aren't you? Gluconeogenesis, indeed. So when you think of a cramp that you've had, if you're busy playing some sport and you need to run again, yes, you take a little while, that lactic acid's going back here, being converted into glucose, ready for you to keep on playing. But you need enough oxygen for your muscles to work. And that's why fitness encourages that activity. Is that comfortable? Answer your question? Okay. Everyone comfortable with that? You look a bit worried. I'm not going to get you to run up and down, unless you want to come running with me in the mornings. It's a bit darkish. No lights out where I live. Can I move to the next one? Yes? Is everyone comfortable with this stuff? Because this is the fed, fasted and stress state, which you've got to talk about in the exam, the final exam. Okay, well, let's turn that glucose loose onto the circulation. This cytosol, whose cytosol is it? Hepatocytes. Liver, please write it down. The cytoplasm, the cytosol of the liver cell, the hepatocyte, what's it doing? It's taking G6P. Where are we? We're trying to go through gluconeogenesis and turn the G6P back into glucose. We're going to let it loose, but we can't get glucose 6-phosphate because it's trapped. We have to take the phosphate off, but the trouble was we went in the liver, we trapped it with hexokinase. Let's go back a couple of slides. Oop, there it is. Hexokinase trapped it. Now it's there. I can turn it into glycogen, turn it into fat or energy. But once I've done this, there's no way back. This glucose 6-phosphatase is the enzyme to take the phosphate off to reach the glucose. Well, here it is here. Glucose 6-phosphatase. It's an enzyme. This yellow strip through here is not some go-kart track. It's actually the ER membrane. Now, we've just seen back here, these enzymes, these characters here, one promoter regulated, this one turns this using biotin. Now we're seeing an enzyme that's stuck in the membrane of the ER. That's an integral membrane protein. That enzyme sitting there, what happens? Well, I have to get glucose into the ER lumen in here. I need this transporter, another integral membrane protein, those glucose transporters we saw before. That was for glucose into the cell. Remember the family members? What about the one in your gut? What's that called? You're all whispering. And its acronym, you're right on target. What are the first letters almost? SGLT. How many sodiums did that drag in? 2. There's no sodium here. It's dragging in glucose 6-phosphate. This is not a glucose transporter like you know it, you've seen before. This is a glucose 6-phosphate transporter. This molecule is coming into our ER lumen selectively. There's no energy required in this case, but glucose 6-phosphatase now is going to rip off the phosphate, no surprise. And here we have this guy, a glucose transporter T2. T2 is going to kick out glucose from the ER into the cytosol. That's dangerous, isn't it? Glucose in the cytosol of a liver cell going through what? Fed, fasted, or stress state? Fasted. So the liver's under glucagon control. Could it be the stress state? Yes, write it down. So that means what's happening is now this liver cell is told whatever glucose is in the cytosol, you're not going to trap it. Why? Because you've just shut down hexokinase. Whatever glucose 6-phosphate going back up here through gluconeogenesis, I'm coming up, but you need to get out, take the phosphate off. What have you got to do? Into the ER, rip it off, send the glucose out, and you're going back out GLUT2. Wasn't GLUT2 the GLUT transporter we saw on hepatocytes last week? Yep. Last week it sucked it in, didn't it? This week it's going to spit it out. That's what's different about the GLUT2 transporter on hepatocytes. It sucks it in when there's lots in your blood. It kicks it out when there's not much in your blood. Does that kind of make sense? So the glucagon's driving that process. Now what you've got here is lots of inorganic phosphate in your lumen of your ER. You've got to get that out. Mitochondria need that. Eventually turn it into ATP. Who else needs it? Well, let's go back here. Isn't this an energy expensive process? And if I don't have any money, I can't do this. That's critical for yourselves. You comfortable with this concept? Yes? So you in the fed, fasted or stressed state now? Let me rephrase that. Stress, let's ignore that for the time being. Are you in the fed or the fasted state? When did you last have a meal? Right now. That's like the Maltesers ad, isn't it? And it rolls down the fan thing on the bench. What are you in now? When did you last have a meal? That's what you ask yourself. When did I last have a meal? When do you think your glucagon levels would rise? How long after a meal? Depends on the meal. That's a good question you'd ask. Roughly 4 hours. If you had lunch roughly 4 hours ago, around midday, right now you'd be going into gluconeogenesis, unlike these characters here are busy going into glycolysis. That means right now your liver is doing exactly this, trying to maintain your blood sugar levels at this point in time. And when you get home and raid the fridge, you'll be in the fed state, won't you? quiz in an hour and a bit. Might be a stress state, mightn't it? Adrenaline's going to do the same. It's going to stimulate blood glucose quickly. Why? Your brain needs it to think. What the hell's the question? I don't know. He's got this really dumb one we don't understand as usual. We done with this one? Okay. Let's look at this control. And I keep talking about fed, fasted and stress because you need to discuss it. On the left-hand side, circle liver. On the right-hand side, circle muscle. On the left hand, we've got glucagon coming in, so that tells us what? Write down which one it is. Fasted. What are you doing? You're turning any glycogen stores into glucose 6-phosphate. Yeah? There is my polymer of glucose as glycogen. I start, remember the big branches, I start ripping them off. The trouble is when I do, I end up with this stuff, glucose 6-phosphate. Isn't that the same thing here? So when it comes from glycogen, we have to cut it off and then still send it through the ER. Not just gluconeogenesis, but all your glycogen stores. There it is, glycogen coming through, glucose 6-phosphate into the ER. Now I make blood glucose levels rise. That means I increase what is termed glycogenolysis. You need to know that term. Now I said at the beginning of the semester, there's some few chemical structures you need to know. The carbohydrate pathway is a key one. Some of the key energy carriers are also key, but I'm using names. So when you search on Google, you can find that for the mid-semester. What you're doing here in the liver is increasing glycogenolysis. You're decreasing glycolysis, you're increasing gluconeogenesis, so you're going to have to fill in a form soon. On the right-hand side, we see adrenaline stimulating muscle. What does your muscle do apart from smooth muscle? Increases blood volume for around the blood vasculature. What's it doing to your cells? It's stimulating their activity. I'm hyper, I'm ready to go, lots of blood glucose. In your muscle cells, your muscles need to be ready for flight, run away, or fight. They need ready to be worked. They need glucose. They've got their own glycogen stores. But why have we got calcium hanging around here? Sorry? You're absolutely right. That is calcium controlled. ATP does the work, little motor proteins we'll talk about next semester, but calcium sends someone out the way so this guy can work. Beautiful little motor proteins. You can actually see them dragging stuff around the cell. It's fantastic. And you can put things on them, a bit like you put on a flea and watch it drag around the cell as well. So calcium, I want you to write next to that, that's a common cofactor. It stimulates, it helps enzymes do work, and in muscle, it helps them contract. It moves proteins out of the way, quickly ready for them to work. Normally, your brain sends an action potential down the neurons, lots of sodium pumps moving stuff out, but just before the synaptic cleft, you covered that in, what the hell is it called now, the physiology unit? You cover the synaptic cleft? Tell me you cover it. Intro to what? You cover the synaptic cleft. You release acetylcholine, yeah? Sodium barrels down here, changes the action potential, but right at the end, as some of these guys know, it's calcium. It says fuse the vesicles, release acetylcholine. Calcium is the worker. It's the guy saying get busy. It's kind of like the raspberry cordial. But that means it works mostly in the cytoplasm. It helps enzymes work. There's your muscle releasing glycogen. What does it need? Glucose 6-phosphate to make pyruvate. So let's go back here. Is your muscle doing this step? No. Make sure you write it down. Muscle glycogen is coming out to make energy, to make ATP. And if you don't breathe enough oxygen, you're going to get a cramp, aren't you? So there's a difference between the fed and the stress state, and now the fasted state and the stress state, isn't there? Differences in how the tissue responds. So your liver, in a fasted state, is busy trying to make glucose. Your muscle, in a stress state, is busy trying to make energy. Your liver, in the stress state, if it has, and I want you to write down, if it has enough ATP, because that's going to be expensive, it's busy making what? Blood glucose. So that fed, fasted and stress state, exam question, there's no reason you can't cover all this stuff, is there? Now, this can take seconds to release glucose, which means these enzymes have to work really quickly. But if you think about being stressed all the time, constantly, every week in this lecture theatre, you are building up the systems to be able to cope with that stress, aren't you? That's what I do, is I train you to cope with stress, to challenge you. Yes, that's what I do best. Are we done with this one? Okay. There's a small problem. The problem is when you use oxygen, you generate free radicals. You also have to manage protons, energy balance. Here you're seeing parts of glycolysis. And remember at the back end where we split the carbons of glycolysis, we had this NAD plus coming in to produce NADH. NADH is a heavy electron proton carrier. That thing can do some damage. It's pretty busy. This is the cytoplasm of the cell. It can be used in some instances to make ethanol. Lots of happy juice to have fun with. You don't make it, you consume it sometimes. But let's have a look over here. What's this talking about? Glucose to pyruvate. I'm using the NAD plus, NADH process as well. But then if my muscle gets deprived of oxygen, as I have over here, what do you produce? Write down lactic acid so you don't get confused. You're not lactating, you're producing lactic acid. Look at the lactate. You see it here, L-lactate. There's 3 carbons. What do you see on this end? What's that? What form is that? It's an acid, isn't it? Lactate, lactic acid. If I have a proton, it's lactic acid. What did you start with? 3 carbon molecule. This thing, I want you to write down LDH, lactate dehydrogenase. We used to do this in the lab in the old days, particularly from liver. It was a pain in the butt assay because the quality of liver to produce this and manage it is difficult. And what does it do? One second. Lactate dehydrogenase can also convert back to pyruvate. See that other way arrow? Is that going to happen in your muscle cell? Well, not so easy. What's its job? Well, if you're depriving your muscle of oxygen, you generate this pathway. That's the cramp. Yes, mate.
Why do we have this notion of lactic acidosis? Because when you compare pyruvate and lactate, H plus is being put onto the lactate through cycling.
Yep. Yeah, look, a lot of the terms are used in common phrasing, don't always make sense. So lactic acidosis often is where you get the acidosis effect. So protons here may be being produced because you have this acid function on the end of that lactate. And you often see people talk about lactic acid, but not actually the lactate. Why? Because you've got protons managed in other ways. That's the key issue for that. So be careful with kind of colloquial terms and what people use out there. You know, scientists now, lactate is the key. In this case, NADH. What does it do? NADH produces lactate. There's the proton to balance the charge. And that's why it's lactic acid. Now, that's going to be painful and sometimes causes damage to your muscle, doesn't it? What do? You send that character back to who? Oh, let's go back here. Back to your liver to basically neutralise it. Send it back to what? And that's why I asked you to write down amino acids when. If you're not stressed. If you need more glucose, oops, ooh, there we go. Trigger happy today. If you need glucose, you need energy, you're going to turn that into pyruvate. Now, what I'd like you to jot down on these slides, doesn't matter if it's this one or this one here, the flux change can happen like that. That's why I asked you to run over to the Building 21 car park and run back again. In a few seconds and minutes, already you have an issue to manage. How quickly does that cramp dissipate? You know, you stretch the muscle, they get you down the ground, they make you stretch. It's fairly quickly, isn't it? I mean, it's painful, but it's fairly quick, which means it's now out of the muscle, heading back to the liver, being processed really rapidly. We done with this one? Yes? All righty. Now we have another cycle to keep in mind. I want you to look at the cycle and see if you can reflect on other cycle that we've seen before that looks kind of similar, but has a different name. Who are the tissues? Muscle and liver. What are we doing? Well, we're busy working and we just produced, write down lactic acid. So you know your muscle's busy working. you're depriving it of oxygen that it needs for the amount of work you're putting on that muscle at that point in time. What happens to the lactate? It goes back to the liver. Under this process, if we say, well, I've still got to play the game, you know, I've got to get up, get rid of the cramp, and I've got to keep going, I might slow down, try not to get another cramp. What does your liver do? It produces glucose. This is the Cori cycle. You need to know the Cori cycle. What does it look kind of similar to? Where you had something cycling from the muscle to the liver and back again. What was the other simple cycle that we talked about? Alanine cycle. They are different, aren't they? This one's moving a 3-carbon lactate right down there, 3-carbon lactate molecule. The other one's moving a 3-carbon alanine molecule. Weren't both muscle cell types busy working? Yes, they were. It depends on the rate each is working. Done with this one? Yes? Can we move on? Okay. All right. The pentose phosphate pathway. Why are we talking about this? Well, just down the bottom here, I want you to note we're producing a ribose. A ribose is used in DNA and RNA. Didn't we see that with our energy carriers? A base, a sugar, phosphate linker. So you can make DNA and RNA. It's often used in coenzymes as well. That's right down the bottom of this pathway. What have we got in this pathway? Within a cell, we've got glucose 6-phosphate, number of steps using NADPH, make 6-phosphogluconate. If I take a carbon off, I've made a 6 sugar into a 5 sugar. Write it down. A 6-carbon sugar up here, glucose, activate it with phosphate. This thing, this transaldolase, is going to help us make a 5-carbon sugar. Already active. That 5-carbon sugar here is ribose 5-phosphate. That is the sugar you see in DNA and RNA. Whatever base, sugar, phosphate linker. That's useful if you're making lots of DNA, that is, cells are replicating. It's busy when cells are also making lots of messenger RNA, so they're busy working, maybe differentiating, which we talk about next semester. The problem you have got is this step here, NADPH to NAD+. This is a lot of energy, a lot of electrons, causes damage. We're going to see it again after the break. You need this enzyme, glutathione reductase. That produces a glutathione, a glutothione, glutosulfhydryl. That's what that GS stands for. It starts life as a dimer. Glutathione, sulfur, they are linked together with a disulfide bond. That's the GSSG thing. This enzyme clips the 2.5. Now I've managed them. Why are they so important? Remember we saw sulfurs being used on who? Who uses sulfur to activate molecules? Where do we see it before? Where, sorry?
ACP.
Yeah, not that unit, no. Whereabouts in this unit? Where have we dealt with it? We use it in ACB, advanced cell biology, to cleave disulfide bonds to get things to work in tissue culture, for example. Where have we seen it here? Who's used a sulfur to activate somebody? Which energy carrier? Acetylcholine. I'll leave that with you to ponder. We'll come back after the break, so you've got time to ponder. This is important for managing free radicals as well. I want you to jot that down because after the break you must use this, because in oxidative phosphorylation, the chances of generating oxygen-free radicals in a high sugar, high fat diet increases. Not only do you put more sugars on immune cells, they get busy challenging you, you have more free radicals, which means you damage cells and potentially have more tumours, which again we're going to talk about today. Time for a break? So you can work out where or who else uses sulfur. Yes, mate.
Where does this happen?
Happens in the cytosol, particularly in liver cells. Just hang on a sec. Yep.
This is such a surprise.
Okay. Just seeing who's awake. A couple of questions during the break. I've been chasing a few demonstrators to make sure they're updating the stuff on views. You all should have your marking sheets back, so we get down the track, and if they haven't updated, just send me an email and I'll give them a hot poker to make sure they do that for you as well. Some of your colleagues are asking about saline. What is saline? And is that all it is? Maybe, that's a good response. Is it or isn't it? If you're going to get a saline irrigation, you're in the hospital somewhere, they irrigate you with saline, that doesn't mean you're going to get sprinkled with it. Like a sprinkler, you're going to put something on. It's just sodium chloride, isn't it? 0.9, what? Weight per volume, what does that mean? You do have to work it out. 0.9%, what does percent mean as WV? I checked that. That's what you have Google for to check, isn't it? Is it per mil or per 100 mils? What's percent? I checked that. Make sure you know absolutely. Now, you're also asked to look at saline in the lab. Saline has nothing else in it except sodium chloride. I mentioned that last week, right? If you try to detect how much protein is in there without spiking it with anything, how much did you see? Hopefully none. If you put a known amount in of BSA, how much should you see? The known amount. If it's not that, then there's interference. For your beverage, we asked you to run a spectrum as well, didn't we? To see if, like, some of these weird things, Fanta, Coke, all the things you guys drink, Mother, whatever it is, does that overlap? Does that screw up the assay? Did you spike that sample to see what recovery you got? Yes, you should have. If you didn't, uh-oh. That's the idea of spiking, isn't it? I went through that last week. Should you show the spectrum of both of those in your presentation? If you're a team leader, that'd be wise, wouldn't it? In your lab book, shouldn't you discuss that with your beverage and the data? I think I would if I was you, because they're going to get tougher and tougher on your lab books now. We're expecting more and more because you're getting hopefully better and better. Or you're going backwards, one or two, because you don't care. That's up to you. Yes, ma'am. When you finish your Malteser. Yep.
Should I put in my lab book the results for all three beverages, or in my lab book?
And what does it say about the team leader? What does the team leader need to do in their presentation?
Obviously, in the presentation, I talk about all
You
three,
summarise.
but
Yep.
should I put all three in my lab book?
Team leader lab book is assessed as a lab book for you.
For me.
Okay? We're only assessing the individual.
Yep.
I hate group assessments with a passion because I had probably the same experience as most of you. So I don't do group assessments. But you still have to work as a team. Resources, getting organised, coordinated, be a manager, all those things you have to do in the real world. Make sure things happen. Should we get back to work? No, you don't want to go back to work? Too bad, let's go back to work. Okay. Why is this pathway so important? Glucose 6-phosphate, there's an enzyme here, dehydrogenase. It's using magnesium. Magnesium's what? Cofactor. With NAD plus to generate electrons and protons. What have we produced? 6-phosphoglucose delta lactone. Oh God, that looks pretty ugly. You know, it's kind of circularized with that oxygen. Then we have a lactonase. In comes water. There's some more magnesium. Cofactor, trace element mineral. What did you produce? This thing, 6-phosphogluconate. Now you need to watch my laser. You need to draw a nice curvy arrow down the bottom there, through there, up there, and then down to there. So join this with that. Done that? Didn't hit the sides and buzz on you? What are you doing here? Well, you've got more, bless you. more magnesium, more NAD+, this dehydrogenase, you kill off a CO2, there it goes, you produce ribose 5-phosphate. CO2 off, 6 carbon, now to 5 carbon. We run down to this isomerase. What have you got now? Instead of this D-ribulose, you've got a D-ribose 5-phosphate. This is the pathway over on the right, nicely summarised for you. The pentose phosphate pathway. I said I'd give you a tickle of it, and that's all. Keep in mind it's important for managing NADH, NAD+, and also for free radicals, but also to produce 5 carbon sugars right down to here for nucleotides, DNA, RNA. Particularly in busy cells multiplying and busy making lots of RNA. That usually means differentiation or repair or immune cells. When they differentiate, mature, they produce lots of RNA, so they need lots of ribose 5-phosphate. We finish with this one? Not quite. Okay. Now, ready? Yep. Everybody else right? Good question. Do you need to know this? I think you'd need to know that there's magnesium used a lot in here. You clip a carbon off with this enzyme, the dehydrogenase. NADH is generated a lot. What have you got? Lots of couple-ended arrows, lots of one-way arrows, one there. You went around to produce a substrate sugar for nucleotides. That's what it's doing. I don't ask you in the exam to give me every metabolic step having every carbon, but what you're doing. You're clipping a carbon off from a 6-carbon sugar. The glucose you ingest is going to be turned into a 5-carbon sugar to make DNA and RNA, depending if you're replicating cells or differentiating cells. And that's kind of critical for growth of tissue. Does that answer your question? Cool. Have that with the Maltesers. Okay. All right, can I move to the next one? Or the banana. Now fill in the boxes. Control of carbohydrate metabolism in the liver. Start at the top, high blood glucose. High blood glucose, what's your insulin? Arrow up or arrow down? Next step, GLUT2. The glucose inside the cell, arrow up or arrow down? Over here, insulin-sensitive protein kinase, arrow up or arrow down? Down here, protein kinase B, arrow up or arrow down? Put them in, don't be scared. It's better to get it wrong and get it corrected rather than not do it at all. GSK, who do you think that is? What was the first bit? Absolutely, good job. Glycogen synthase kinase. It tells you down here, what's it going to make? Glycogen. Over here, PP1, didn't we see that last week? Is that up or down? The next row, glycogen phosphorylase, is that up or down? Let's have a look. Did you get them right? Fill in the boxes correctly in case you didn't. Make sure you get it right, because you might find an exam question ask you the kind of things like this. Which one's up, which one's down? What does it mean by up and down, by the way? Increased or decreased in activity? Increased or decreased in activity. Why? Because you are under high blood glucose. That's in what state? Write it down. That's the fed state response for your liver cell, muscle cell. What's making glycogen? There's glycogen synthesis going up. It's increasing glycolysis. That's the way to go. Glycogen breakdown is being decreased, isn't it? Glycogen breakdown decrease, which means you're not breaking up those branches to make glucose. So you inhibit that process and you enhance the glycogen synthesis process. on glycogenin. Remember that little protein that seeds the process? Are we finished with the top half? Not yet. Take your time. This is a liver in a fed state. Could be a high glucose meal, and I want you to jot down it could be a high protein meal, does the same thing. When you're ready, start the bottom one. And don't wait for me. Put in the arrows. It's better to get it wrong and work out why than do nothing. So in down below, we have low blood glucose. What states that? Fasted. So your glucagon should do, I think I can already see what's underneath there. Should go up, shouldn't it? What about cyclic AMP? Remember, this is signal transduction. GS-alpha, adenyl cyclase, cyclic AMP, key second messenger, would that go up or down? Should you produce more of it in a fasted state or less of it? I think I can see what that's doing. That changes protein kinase A. I can already see that. What does that do? Phosphorylase kinase, is that up or down? Jot it down. Should I come around and check all of you? What about this stuff? PFK2, is that up or down? FBPase, is that up or down? Pyruvate kinase, remember that character? Is that up or down in the fasted state? This is a fasted state, isn't it? F26BP, remember there's that part of glycolysis or not? Yes or no? No. PFK1, should that be up or down? What are you doing in the fasted state? Low blood glucose. What does the liver have to do? We're in the liver cell now. Make glucose. Glycogen synthase. Should you be increasing that? I think I can already see the answer. What about glycogen phosphorylase? Let's look at the answers. What should you get? Make sure you fill them in correctly. Doesn't matter if you got them wrong the first time, but it does matter to get them right this time. And ask yourself why. What's going on in this cell? So in that short answer question, apart from a bunch of multiple choice that you have to barrel through in the final exam, these are the things you should be discussing. Who goes up, who goes down in the fed, fasted and stressed state? After the break, we're going to see the hormonal activity, what your gut's doing to your brain. We done with this one? What your fat cells are doing to your brain. And you'll have to trust me, they do things to your brain. I know you think, no, they don't. Yes, they do. Can I move on now? Has everybody checked this? They've got it all done? Chris, got it all done? Cool. Anybody else? Can I move on? Sure? Maybe if I stay here for a while, you'll be more comfortable. Let's look at all the overall balance. ATP synthesis from glucose. How many glucose molecules are we going to start with? Just one. Overall, what we have is direct products, NADH, a couple of those, ATP, a couple of those. Remember those inputs and outputs. NADH, or mitochondrial matrix. Why is that? We'll come to that after the break. NADH, mitochondrial matrix, after the break. Cytosol we know about. FADH2 and some ATPs or GTPs. Just write down GTP, its equivalent as ATP. We'll talk about that after the break. You'll notice the overall final ATP, 3 or 5, there's an asterisk there. 2 ATPs, 5 NADHs overall, 15, 6 NADHs, 3 and then 30, 32. Now why 30 or 32? Because it depends on your inputs and your output. If you go back to fats, a 16 carbon fat generated how many do you recall ATP equivalents? A 16 carbon palmitate fat. Do you recall how many ATP equivalents we got out of that? It might help you to remember a glucose is roughly 30 or 32, 6 carbons. A 16 carbon fat is 130 odd ATP equivalents. So one fat molecule, 16 carbon long, generates another 100-fold more ATP or energy equivalents. So it's a very energy efficient process, that is fat breakdown. Keep that in mind. So glucose costs a bit. Trouble is, this thing doesn't use fat, does it? So you need a lot of glucose to keep that thing busy, or vice versa. We're finally done with this one. So yes? Okay. This is where it gets more interesting. And after the break, we start getting really into the guts of what goes on. I mean your gut. How your gut tells your brain to work, how your fat cells tell it to work. Here is a tumour mass. In the lab workshop this week, your will assay is based on PEP, phosphoenol pyruvate. You're going to get the information that says the sample was derived from a tumour mass, an amount of material, wet weight. You'll have to do the PEP assay and convert that back to an amount of PEP per amount of wet weight. Please take note of the instructions when they hand them out. You've got to convert back to a weight. What happens in a tumour mass? Some of the advanced cell students know this. If you do some other units, you'll hopefully get to cover it as well. A tumour starts off life and it might overcome cell cycle regulators, so it starts to get out of control. Instead of differentiating and doing things like it's supposed to make you, it says, well, I'm going to do my own thing, which might be a mutation, it might be a virus, it might be anything, it might be a drug, alteration in activity. Key regulators running around the cell cycle. So now instead of running around and say, no, I'm going to stop in the S phase and differentiate, I'm just going to keep running around. That means I'm going to keep on dividing. So the brakes are off, what we call apoptosis, cell death. How do I keep running around? Well, the trouble is in a mass of cells, I have to differentiate myself from the other cells. Tumours do that. That's why they wall themselves off. They wall themselves off to a tumour mass size. What do you think the overall maximum size you can get a tumour? Before you have to see this process, I want you to write down angiogenesis. How big do you think a mass of tumour cells could get before they have an issue, before they need to induce angiogenesis or metastasize? What size do you think they could get to? Women should know, because often you have to have mammary checks, breast screening. What's the rough size they work on? Sorry? It's a bit big. On the right track. How big do you think? Come on, think about it. Sorry? Bobby head, what's that? Pin. No, that's a bit small. Let's find the middle ground. It's about 1 centimetre. Write it down. About 1 centimetre, then you have diffusion issues. The centre of that centre mass of cells can't diffuse the good stuff in and the waste products out. But tumour cells wall themselves off. While they're doing that, they're trying to get busy. What are they doing? Making lots of DNA to replicate. What do they need? Lots of energy to replicate. How do they get into this process? Well, they take glycolysis and they increase the enzymes to make more ATP through glycolysis. We can see that. I can show you in advanced cell how we use things like NADH to tell an increase in activity of tumour, particularly in skin cells. They increase their glycolytic activity. How do they do it? They increase the enzymes required. Here's a tumour cells increasing more glucose transporters. So not only do they increase the rate of glycolysis, they put more glucose transporters on the surface to suck glucose in. That's what tumours do. Get down here, they increase the amount of hexokinase. What does that do? Traps it in the cell. So a tumour cell not only sucks in more glucose, it keeps it there. It increases the amount of all of these proteins. It runs down and it increases all of the glycolytic enzymes. Is it going to make glucose? No. It wants to make lots of energy to replicate itself. And that's what a tumour does in the first few stages. Before it starts inducing, and that means another change in its activity to induce blood vessel growth into it as a mass to supply oxygen. Even then it's still demanding to get rid of the waste products as well. This process means that these cells need more glycolysis and also, we'll see after the break, more oxygen. And you can manage some of these cells. Hamad just did his COC this morning. We're managing them from the electrical activity in the nucleus, altering DNA activity. Here is a tumour mass now that is being stimulated to grow well. Why? Because you've walled it off, you don't get enough substrate in. I increase all of my enzymes, my proteins to suck it in. What do I deal with? This thing. I've written it down here for you. HIF, hypoxia-inducible factor. What is hypoxia? Low oxygen. You can induce that in yourself, couldn't you? Just turn down very slowly the amount of oxygen in this room. You'll be inducing those enzymes that generate more ATP without as much oxygen. You'll see that after the break. Only the proton 4 complex actually uses oxygen. So here we see tumours increasing glycolysis. That's a common activity of tumours early on. What kind of proteins do they increase? Particularly through this HIF factor, GLUT1, GLUT3, hexokinase. What's phosphofructokinase? What's its acronym? PFK1, write it down. Aldolase, one of those other key steps in our pathway. Glyceraldehyde 3-phosphate dehydrogenase, phosphoglycerate kinase, enolase, pyruvate kinase, lactate dehydrogenase. Lactate dehydrogenase, that's interesting. Didn't we see that a few slides ago? So a tumour mass goes into up-regulating glycolysis. Really busy, lots of energy to do the work it needs to replicate. That's what tumours do. If they're going to differentiate and induce blood vessels, it's a different step. Why we're looking at this stuff is because some of the metabolic pathways you've selected for report part A is about an enzyme or a factor that's altered. I'll talk about more of that in the Q&A tutorials. Report part B, you're seeing some of these characters. Compounds, D-glucose. We use this in advanced cell. It screws up glycolysis. It competes for glycolytic pathways and enzymes. It shuts down glycolysis. So D-glucose is often used for tumour treatment in the early stages. Here's another one, linodime. That screws up the pathway as well. 3-bromopyruvate. That is a chemical agent synthesised to screw up a metabolic pathway. That's report part B. When you read the details, you'll see that I've embedded both elements. Part A is about a genetic disorder, a mutation, some factor not working correctly in part of the process. Part B, who screws it up? Like these compounds, these synthesised molecules, maybe from a plant, maybe chemically synthesised, that are drugs, metabolic manipulators to alter metabolic pathways. Does that kind of make sense? We see some more down here. Oxythiamine here screws up some of this pathway as well. What's that trying to do? Manage a 5-azylose phosphate. Here's our HIF. So low oxygen increases this pathway, increases glycolytic enzyme. So as the tumour walls itself off, and that's different to a cyst. A cyst you can move around. A tumour walls itself off, you can't move it so easy. And if you see something about 1 centimetre, you know it's about maximum, and it might get bigger if it differentiates and forms blood vessels. Or it starts metastasising. Those are differentiation steps tumour cells have to take. Hypoxia is going to be important for the mitochondria. Again, that's after the break. What we have to worry about is too much glucose inducing free radicals and inducing tumour formation. That's what we have to worry about. We done with this one? Okay. What about our pyruvate? What do you see over here? Glucose, gluconeogenesis, OAA, there's our pyruvate carboxylase, there's our PDH complex. Write it down. PDH. I want you to put an arrow down all the way saying glycolysis so I can get to pyruvate. If I get told to make glucose, I go back up. But once I get to pyruvate, I have to make a decision. Am I in the fed, the fasted or stressed state? What is your liver going to do? Depends on the signals it gets. Write down PDH is that big enzyme we're going to see in a moment. PDH is going to turn pyruvate into acetyl-CoA. Pyruvate is in the cytosol. What I want you to do is draw a little line there. I'm going to take pyruvate into the mitochondria. So underneath where my laser's zooming across, big circle, write mitochondria. A mitochondrial matrix. PDH is in the mitochondrial matrix. You saw that spinning cup in that Harvard animation, hopefully you recall. PDH is going to take pyruvate, a 3-carbon molecule, put 2 carbons on CoA and generate CO2. That acetyl-CoA could enter the citric acid cycle to make energy. Is that the fed or the fasted state? Depends on the cell, doesn't it? If that acetyl-CoA comes back out of citrate, what are you making? Back out of the mitochondria? You're making fat, aren't you? So you'll be in the fed state. This acetyl-CoA, if it builds up, square bracket, cars on the lot, it feeds back to PDH and says, stop making acetyl-CoA. If you turn acetyl-CoA to fat, it'll keep on making it. Who does that? The liver. If that acetyl-CoA is being used for work in your muscle cells, then you keep on making acetyl-CoA. Make sense? Hopefully does. But that acetyl-CoA also has a positive effect on pyruvate carboxylase. It builds up. It says, we're done, thanks. Let's go back up, turn it into glucose. What are you doing with all this? It depends on whether you're storing carbons or you're burning them. Can I move to the next one? One second. Now, PDH is a complex enzyme. It is multi-subunit. Remember we saw the biotin handing off from someone to another active site? Well, PDH is similar, but not just 2 active sites, it's got a couple. Can I move to the next one now? So yes? Okay. Here it is here. You can see it in an EM. It is a monster. It's 4 to 10 million Daltons. It's huge. That's an EM showing you this character. Here's the space-filled model. Notice there's E1, E2 and E3 sites. I want you to think about E1, I start, I then transfer it to E2, and then I take it to E3. Remember we saw in that biotin, we saw the lipo arm, that long chain carbon that grabbed from one site, swung to the other. This is very similar. A lipo arm again, once we design it, we can modify it. PDH complex, big multi-subunit, 3 different kinds, multiple copies, 5 different cofactors. TPP, FAD, CoA, NAD, and lipo8, which is our arm swinging around. What are you doing? Well, in the mitochondrial matrix, you're going to grab a 3-carbon, clip off a carbon, generate acetyl-CoA. That's a decision. Once you do this, once pyruvate's in, you'll see a transporter taken in the mitochondria. That's it. We're either committed to make energy or we're committed to make fat. There's no going back up to glucose at that point. So even if glucagon comes along, we're stuck. Acetyl-CoA has got to be dealt with in another way. The decarboxylation is occurring in the mitochondria. That is irreversible. Please note that. CO2 you're exhaling is part of that. Our NADH gives a proton. We go from a 3-carbon pyruvate to a 2-carbon acetyl. Write it down here. 2-carbon acetyl. CoA is just the carrier. Here's the overall equation. I suspect you might get an exam question on that. Can I move to the next bit? So yes. So here he is. Now I want you to look at the overall equation first. Pyruvate, there's the purple carbon, pink bit we've covered. Here's all the incoming cofactors, co-contributors, CoA carrier, outgoing NADH. Here is each of the E1 in yellow, E2 right on the edge, E3 component of it, E4 and 5. Now what does that mean? These subunits do each job. The yellow, green and purple are essentially E1, E2 and E3. Yellow, E1, green, E2, purple, E3. They are the major active sites for each enzyme. Yellow, E1, green, E2, pink or purple, E3. Let's go back one. There it is there. E1, E2, E3. So what goes on first of all? Well, incoming pyruvate, have a look at it. 3 carbons. What do you do first of all? Well, first of all, you grab thiamine pyrophosphate. That's what TPP stands for, thiamine pyrophosphate. What did I do with that? Well, with that, I clipped off a CO2 from the acid component. And what did I generate? TPP linked to a 2-carbon, circle that. That's 2 carbons from my pyruvate. One of them I'm going to exhale. Immediately on that one active site in step 2, I transfer it to this blue thing. That's the lipo arm. There it is. I've taken the 2 carbons of pyruvate and I've stuck it on there in the E1 site. I've stuck it on there in the E1 site. Oh, there's sulfhydryls. Haven't we seen those before? It's kind of like phosphate, but it's a weaker activity. What have I got? A disulfide bond, and I've used it now to generate a sulfur that now has 2 carbons of pyruvate on it. I swing it around the arm. That's what the arrow is here. I swing it around a lysine, so my lipo arm, I am lysine. My arm is the lipo arm. Swing it from E1 to E2. What happened there? CoA came in. There's the sulfhydryl. What did I produce? Acetyl-CoA. Just circle it. When I did that, I ended up with this sulfhydryl. There's my hands, now a sulfhydryl function. To get them to work, I have to reactivate them as a disulfide bond. Who does that? E3. Let's think about it. E1, I clipped off the carbon. I put the 2-carbon pyruvate component onto a lipo arm. E2, I transferred the 2-carbon to a CoA. E3, I just generated the disulfide bond again. Does that make sense? This is an unusual enzyme in that context, but it's a big character. Use the overall equation for that reaction. Incoming 3 carbons, outgoing CO2, oxidation, generate my acetyl function, transfer to the CoA, there's my acetyl. I can turn it into fat or I can turn it into energy. What did you make with a long chain palmitate? Beta oxidation, then you produced an acetyl-CoA, didn't you? We're going to see that after the break, how you manage it. Are we comfortable with this enzyme? There will be a few questions on PDH, I'll guarantee it. Yes, mate. I think I'm getting you. So E1 is the yellow area shaded. E2 is the green area. E3 is that purpley pink area. E1, you are grabbing pyruvate and ripping a CO2 off, but as you do it, you transfer the 2 carbons of the pyruvate, these characters, and you put them on the TPP bit in the active site and immediately transfer them to this disulfide bond, and you end up with 2 carbons linked to a sulfur. When you do that, you break the disulfide bond and end up with a sulfhydryl or 2 carbons on pyruvate. That process in E3 transfers those 2 carbons to CoA. There's your acetyl-CoA. That's why it's colour-coded. That's why I like Leninger so much. It actually helps you see what's going on. The trouble is, overall, I've ended up with 2 sulfhydryls. They can't do anything for me. I need to regenerate the disulfide bond, which is what happens in E3. I regenerate the disulfide bond, all swinging around this lysine amino acid on this lipo arm, drag it around, there it is, disulfide, put a pyruvate on it, 2 carbons, swing it around, sulfide, there it goes, acetyl-CoA, regenerate it. Does that make sense now? Everybody else right? Okay. This is grunt biochemistry, but what it shows you is how an enzyme can work rather uniquely. Multiple active sites doing different things, which is a step up from our biotin, wasn't it? 2 active sites, one here, transfer over to there. Simple. Yes, mate? And don't say sorry.
I don't really understand the big picture of this. Like, what's it? Why and what's it doing? Like, what is PDH? Really, I'm not following.
Okay, let's go back a step. This process is pyruvate, right? We've spent the last day and a bit, lecture time, taking glucose down to pyruvate, right? Even fat. You're going to make fat, you have to go through glycolysis to make pyruvate. That pyruvate molecule, once I've generated it, I could go back up if I'm in the fasted state. That's the decision glucagon tells me to make if I'm a liver cell. But if I'm going to turn it into energy and generate the citric acid cycle, generate lots of these electrons and protons, what are they going to do? They're going to pump protons into the intermembrane space of my mitochondria. What do? They all line up. How do they generate ATP? They come back in through ATP synthase. So this process means I've got to take a 3-carbon pyruvate, transfer it with PDH, 2 of those carbons to acetyl-CoA so I can enter the citric acid cycle. Once I enter the citric acid cycle in the mitochondrial matrix, I zoom around here, cut off more carbons, I generate a weak energy carrier, which I'm going to ask you to write down as a ATP equivalent. Down here I generate a lot of electrons. Those electrons pump the protons into the intermembrane space. I've got to run through this citric acid cycle to generate those guys, to generate a proton motive force to spin the wheel to make ATP. To do that, I've got to take a 3-carbon and turn it into a 2-carbon. And that's got to be in the mitochondria linked to CoA. Does that make more sense now? Which is probably the same issues most of you have. Why does this have to happen? Why the hell do you take a 3-carbon molecule, write it down, 3-carbon, down to a 2-carbon? CoA is just the carrier, remember that? Because the system doesn't work to run into the citric acid cycle, or taking that acetyl-CoA out of citrate. Citrate, remember, you turn it into fat at some point, don't you? Through the citrate shuttle. And this enzyme says, I'm going to do that, and this enzyme, remember, is irreversible. Once I make that acetyl, I can't go back up glycolysis. I'm in the mitochondrial matrix, that's it. 1 carbon's gone, I'm done. I either turn it into fat or I turn it into energy, one of those 2. Make sense now? Yep. Okay, can I move on? Oh boy, it gets even more complicated. Write down feedback, either inhibition or enhancement. That's what the red crosses are here. Red minuses, I should say, for this one. Feedback inhibition, and the green pluses are enhancing. Let's walk through it. PDH regulation is under stringent controls. Shouldn't that be the case in your liver? If I'm turning this molecule, pyruvate, into fat or energy, it should be very regularly controlled. Who does it? Here's PDH, nice big monster. What do? You put a phosphate on, what do? You inactivate it. Look over to the far right. Phosphate on, inactivate it. If I take the phosphate off, what do? I activate it. What does it do? It takes pyruvate, turns it into acetyl-CoA. All these arrows aren't some flight plan for Qantas or Jetstar. It's telling you if any of these things build up, they either have a positive effect, enhancing the kinase, in the case of NADH, or inhibiting it. Too much pyruvate. Too much pyruvate, what does this PDH do? It should get busy making acetyl-CoA. These, hopefully, and I want you to follow them around. After the break, you're going to tell me what's going on. But what I want you to notice is calcium. What are you going to do about calcium? What are you going to write next to calcium? Common cofactor. Write down in the cytoplasm, particularly. If a muscle cell gets told to get busy, a liver cell gets told to get busy, you see lots of calcium. When an egg gets fertilised, it's calcium. Calcium is that common factor, so if calcium comes out of its storage, everybody's busy. And if everybody's busy, Should you be inactivating PDH or getting it to make energy? Getting it to make energy, wouldn't you? So calcium, I want you to jot down, overrides. That's why it's a potent activator. As soon as the cell's told to work, all these other inhibitors are overridden. Write it down. Calcium overrides the other characters. Get busy, work. That's what calcium's saying. Now, it'll take a little while. If you're doing PASS, I'd encourage you to work your way through it, but just follow the arrow, whether it's plus or minus. Kinase, shut it down. Phosphatase, turn it on. It actually makes a lot of sense. Think about what you're going through, fed or fasted, but as soon as you get a signal to get busy working, all bets are off. Off you go. We done with this one? Positive means it's enhancing. The minus arrow means it's inhibiting. Okay? Like our green triangle, positive enhancement. Red cross, negative inhibition. Can we move on? Oh man, it's a long day, isn't it? This process is termed differential regulation. Regulation of the kinase is key to overall regulation. Overall. Why? Because the cell's being told work, store fat, store glycogen depending on what kind of activity you're in. These processes, now I talked about allosterics, relaxed and tense. When you put a phosphate on, write down it's a covalent modification, a covalent bond. Kinase links a phosphate to somebody, it's a covalent link. What did it do to PDH? Phosphate, shut it down. What are you doing with a covalent intervention? Your phosphorylation of E1. What did E1 do? It grabbed pyruvate, grabbed it, ripped off a carbon. So each one of these steps is intimately linked to E1, E2 or E3, depending on where it's working. Here's the feedback. Didn't we just see this a while ago? Yep, it's the same deal. But now you're seeing the details of why you must do that. Now, these are all related molecules because, as I said, once you commit to this, you've had it. And our calcium is a key character. And you'll notice here, hormones like vasopressin. We'll see that after the break as well. More hormonal integration of metabolism, how your cells are told what to do. Down here, you're seeing insulin stimulates dephosphorylation of the PDH complex, particularly in fat cells. Adrenaline stimulates activation in heart cells. What do you do when you get an adrenaline rush? Bump, 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 bump. What are you doing? Pumping more blood around, more oxygen, more CO2 being pumped out, more glucose being pumped around. Are we almost done? Must be? Why is that? 6 minutes to 6. Yes, we are.